So a few people trickling in here, but we'll go ahead and get started to stay on time. Thanks everybody for joining us today on day 3 of the showcase. Before I get started and introduce the presenter for this talk, just a logistical reminder that you can put your questions in the chat box or the Q&A box at the bottom of your screen. And then Isaiah will answer those at the end of the presentation. So as your questions come up, put them in there and we'll hold them till the end. So it's my pleasure to introduce our presenter here, Isaiah Tolo. He's a third-year PhD student in the Conservation Sciences graduate program and has been working on koi herpes virus in common carp, a project over the last 3 years since he's been here. He's going to give an update today on where he's at and where things are going into the future. Take it away, Isaiah.
All right. Well, thank you for that introduction. I'm going to be presenting on our work looking at viral biocontrol of common carp. And I promise this is my only quip about coronavirus from here on out. Let's see here. So common carp are not native to the United States. They were introduced about 100 years ago, predominantly by the U.S. Fish Commission, which was the predecessor to the U.S. Fish and Wildlife. And they were introduced primarily because they're interesting as a food fish. And they didn't know as much about invasive species impacts at the time. But they quickly realized, but it was too late because carp had already spread throughout the United States and become naturalized. And that means they're basically the worst kind of invasive species. They don't need any help to establish themselves, and they've really got their hooks into the environment, are able to stick around. And this is not just true for the United States, but around the world. So you can look at this bottom map on the left-hand side showing the CABI sort of locations for where carp have been introduced. And you can see it's basically global, though they're still really used as a food fish in a lot of countries. So they've only been designated as invasive in a few different continents, different countries, I mean. And in the United States, on this right-hand map, you can see these clustered specimen records, which is kind of a proxy of where carp are most dense in the country. And they're really dense in our area and off onto the East Coast as well. So the problem with carp is they cause a shift in shallow lake ecosystems from a clear to a turbid water state. And they do this by bottom up and down and top down effects. So not only do they eat zooplankton and benthic invertebrates that feed on algae, but they also disrupt vegetation, which directly competes with algae. They also exert their bottom-up effects, which is their excretion. So these really high biomass populations excrete a lot of nutrients into the water column, and they also stir it up from the lake bottom by their kind of interesting feeding method of rooting around on the sediments. And that causes that algae to over-dominate the system and then and shift to that turbid state. So there's a few different tools out there for controlling carp that are pretty effective for removing large amounts of fish from one system. There are like typical methods like seining, netting methods. There's also electrofishing, is basically the same thing in a lot of ways. And there's also poisoning, which is kind of like to restart a lake. If it's just gotten way too out of control, you can just poison it and kill everything. And also there's some interesting methods of using biocontrol, so using like bluegills that are predators of carp eggs and larvae. But all of these have limitations, like the size of the lake can really make the labor or the cost prohibitive. Also poisoning and biocontrol are not limited to carp, so they actually impact other native species as well. So there really is a need for integrated control methods. Carp's effect on shallow lake ecosystems is really well documented, and removing them might be the only way to get back to habitat recovery for some of these systems. But carp are so widespread, and so it makes it very difficult for us to control them. And programs that aim to reduce sort of their threshold, their density below a threshold where they cause damage could be achievable, but you need to have a pretty high degree of effectiveness. And modeling of integrated management plans is not very promising unless you're also implementing a wide-scale control method, such as biocontrol being one example. So pathogen biocontrol is one type of biocontrol. There's a lot of different definitions for what biocontrol is. Pathogen biocontrol falls into this definition by basically trying to reduce the density of a pest species by introducing a pathogen or stimulating natural occurring epizootics. So you can either put a pathogen in there that's never been there before, or you can find something that's present in the system and try to augment it. The most popular example of pathogen biocontrol for vertebrate species is in Australia, the use of myxomatosis and RHDV to control rabbit populations. And they were able to knock down rabbit populations by introducing these exotic pathogens into the system when rabbits were really dense. And it did reduce them below the threshold density where they caused damage. But as you can see, those rabbit populations can bounce back, and you actually need other control methods or new pathogens in order to continue to knock down that population. So that's the taking an exotic pathogen and tossing it into a totally naive approach. These types of pathogens have to have really high host specificity, so we don't want anything that's going to impact any other species. We want them to be highly pathogenic, so have a really strong mortality effect. And then also something that I'm really interested in, and our project has been really focused on, is finding things that are endemic. So it can be really difficult to go through a regulatory process of introducing a new pathogen. That's kind of a risky scenario, but if we can find something that's already here and augment it, that can be a lot safer and potentially might be able to get that started sooner. And this is actually really widespread use in the United States for controlling pestiferous insects in the agriculture setting. This is what's called an augmentative or conservation approach, where you're looking for bacteria, fungi, nematodes, viruses, pathogens in the environment that are already there, and then trying to encourage them to control a pestiferous insect. So the use of entomopathogens is pretty widespread and really common in the United States. And it works basically by understanding the ecology of your system really well, trying to do things like maybe reduce soil disturbance and enhance those pathogens in different ways so that they can sort of control your species of interest. But there are no really good examples of this in aquatic ecosystems to control AIS. But it's possible that we could apply the same sort of strategy to CARP. So understanding how they fit into their ecosystem and then finding pathogens that specifically target them. And unfortunately, we don't know much about the potential biocontrol agents that are out there for CARP, but we are interested in some viruses, since those can be basically, they're already kind of naturally programmed to only infect one species. So the foundation of this augmentative approach, we started in 2017. Basically, we would look at mortality events of native fishes, so just kind of anything mixed in a native fish mortality event. And we'd also look at mortality events that were just of carp. And we could screen tissues from those fish from different approaches. We use next generation sequencing to find new pathogens. This is pretty difficult to analyze, so we're still in the process of understanding that data. But we also had a more rapid approach of targeting particular pathogens of interest with PCR and RT-PCR that would detect known pathogens like koi herpes virus, carp edema virus, and spring viremia of carp virus, which are all pretty specific to carp. And we could also use culture assays to isolate pathogens. And the idea was to exclude anything that we found in native fish, anything that we found that was in native and carp, and then to identify those potential biocontrol agents that were only found in the carp mortality events. So this is just kind of a map showing where those were around the state. The black ones was really interesting. We found a lot of carp mortality events, so almost 30 of them up until 2018, and we've surpassed that in 2019 and 2020. And then kind of looking at these carp mortality events, we found koi herpes virus in the majority of these, which was really interesting because we'd never found koi herpes virus in Minnesota before, and also carp edema virus, which was the first time that had been detected in the country. We didn't find any spring viremia of carp virus, but we did find co-infections of KHV and CEV, which is really interesting because no one had ever seen that in the world so far. So focusing on coy herpes virus, it's an OIE notifiable pathogen, which just means that it's internationally monitored, and it causes a really severe disease in carp that basically causes shock and death in a high number of individuals in aquaculture settings, and also in wild settings we're finding. It was first detected in the late 1990s in New York, and since then it has spread to 17 different states throughout the United States, and even more up in this last year. And it can cause a high level of mortality. So Lake Billisby in 2018, we estimated that about 4,000 carp died in that mortality event, which was up to 32,000 pounds of fish. So just putting this in context of how much it would cost to do a removal of this scale, it could be as expensive as $10,000 to do this type of removal using a seining strategy. Carp edema virus is also notifiable worldwide, and there's less known about this virus because we're not able to isolate it and study it in the lab setting. But this co-infection of KHV and CEV in Cottonwood Lake, the mortality estimate was anywhere between 1 to 10,000. It wasn't as precise of a mortality estimate, but that could be anywhere between 8,000 and 80,000 pounds of carp that died in that lake. So this isn't to say that these mortality events are always high mortality, but just to see that they can achieve this level of mortality in some cases without any sort of augmentation is really promising. So it kind of looks like we have these two controls for biocontrol of carp, but just to kind of cut to the chase, coy herpes virus is much better studied, so we know a lot more about it than carp edema virus. And so even though carp edema virus is still on the table and we're still looking for it in naturally occurring mortality events throughout the state, we're really focused on coy herpes virus. So the next step was to isolate coy herpes virus. It's a necessary step in order to understand our strain that's local here in context of the ones that have been found throughout the world, and also to have more control over it so we can actually study it in a controlled laboratory setting. Unfortunately, it's really hard to isolate this virus, and it had never been done to isolate this virus from wild populations of carp before. So we took this kind of unique approach of collecting fish from the wild population and then screening them to find some positives that we could keep in a laboratory setting and manipulate their water temperatures in order to get them to shed more. Then we could use their excretions or their tissues to infect naive juvenile carp, which are much more susceptible to the virus. And so we could get really high copy number infections, really high viral load that could be used for virus isolation. And this was successful. So in Lake Elysian, there was a high prevalence of the virus. So we captured adult fish from that lake. This is just sort of what that pathology looks like in panel B and C. And then up in this top right corner, what we're looking at here is actually common carp brain cells that are used to culture the virus. And the cells dying in this picture is showing you sort of what that looks like when these become infected. And then we confirm with PCR how many viruses are there, and we're able to kind of use this to propagate and store the virus. And this is just a picture of a secondary case in one of those juvenile carp, just showing the pathology when it became ill. So being able to isolate this virus is really exciting because it allowed us to start exploring the disease in a laboratory setting. So one of the first things we wanted to do was to look at the species specificity of KHV. This has been done before in other studies, looking for KHV DNA in non-target species that were either experimentally infected or cohabitated with carp that were infected. But the problem with this is that KHV DNA doesn't mean that the virus is replicating. So we don't know whether detecting the virus in those fish that don't have any sort of disease means that they're actually infected. So we use a secondary test that looks for an intermediate step called mRNA that really tells us whether the virus is replicating or not. We decided to start with two minnow species. So we took one minnow species called fathead minnows that are really common in Minnesota, and they're endemic to North America. And then we also looked at an old world minnow, which you're familiar with, goldfish. They're sympatric with carp and where they came from. So that means they live in some of the same places where carp originated from. And there's some question about whether they are susceptible to KHV. So we use them as well. So this is kind of a busy slide, but I'll walk you through it. Starting with the top left, this is a schematic showing the way that we conducted this disease trial. So basically we take tissues from wild carp and infect juvenile carp that were disease-free. And those are the ones with the blue dots here. And then we would move those into a tank with a group of susceptible disease-free carp. So these are another group of juvenile carp with the yellow dots that were not infected with KHV, but they kind of act as a within tank control. So if there's transmission between these groups of carp, then we know that KHV has been transmitted, and we would expect it to also impact the species of interest, which were the fathead minnows shown in the red dots and the goldfish shown in the green dots. And then moving over to this next panel, what we're looking at here in A and B is these lines are showing the mortality of fish. So this is when fish are actually dying off in the tank, and the dots are showing the viral load. So you can see that the initially infected carp are already come in with a high viral load, and they all die off really quickly, and they transmit KHV to the other group of carp. So the positive control works. So we see them getting transmission, and then all of those carp die. But what we don't see is any mortality of the fathead minnows or of the goldfish. But we do see one dot here of one single fathead minnow that tested positive for KHV DNA. So we repeated this trial just to make sure that we could see the same thing again, or whether we wouldn't. And we saw sort of a similar pattern, but we didn't actually see any KHV DNA in that. These black dots, by the way, are showing the viral load in this tank filter. So this is actually as the KHV cell viruses or infected tissues are kind of coming off of those carp and getting entrained in the filter mesh. So we were detecting some contamination, which is what we expected. So to check whether that fathead minnow was actually contamination or a real infection, we did that intermediate step test to look for whether the virus is replicating. So what you can see is that the moribund yellow dot carp were all had replicating infections, but in the fathead minnows and goldfish, we didn't find any replicating infections, which means that they're likely not can't be infected with KHV, since they can't be infected even in a really close quarter setting like this one. Another thing that was nice about this trial is that it kind of let us look at the disease process of KHV and carp. Kind of what happens between carp getting a new infection to the point where it actually dies. What does that disease process look like? So I won't go into all of this, but it's really interesting because it's useful for disease modeling, and that's something that we hope to be able to do with this information. We were also interested in extending this investigation into an actual wild population. So knowing that Lake Elysian was a really good lake to study with the high prevalence of KHV, we went there and took samples of carp and native fishes at different times throughout the summer when KHV is spread. So we focused on common carp as our positive control. We sampled a number of different native fishes, but we did focus on bigmouth buffalo, since they have a really high density in that lake, population density, and they also share some similar ecological niches with carp. So they're a great species to kind of act as a signal for whether there's transmission going on. And then this panel down here on the bottom left is showing the temperature profile of the lake throughout the summer. This green and purple line are showing when we saw buffalo and carp spawning in the lake. So you can see those are overlapping. And these gray lines are showing the times when we sampled. So you can see that we sampled at these different times throughout the year, and some of them occurred when carp and buffalo were both spawning. And we found that in the carp, about a third of them were infected with KHV. So they actually had KHV viruses in their tissues. And 15% of them were shedding. So that was actually great because it showed us that our positive control worked. KHV is there and it's being shed, and there's an opportunity for transmission to occur. But we didn't detect KHV DNA in any of the native fishes, including buffalo, which means that even though there was opportunity for transmission, none happened, which is a pretty good indication that transmission is not happening in these wild systems. We also wanted to look, looking back into the lab, we wanted to understand a little bit more about how KHV is actually transmitted between fish in the wild. So we did a trial where we were looking at whether KHV is primarily transmitted by direct transmission, so fish actually like touching each other, or whether it's transmitted by KHV being spread into the water column and affecting other fish that way, which is an assumption that's in the literature, but has never actually been tested. And what we expected was that transmission was probably most common as direct transmission when fish were asymptomatic. So they're not really feeling sick, but they have a viral load. And so they're coming into contact with a lot of fish socially and transmitting the virus. And that indirect transmission was, if it occurred, was probably happening more during the symptomatic period when fish are feeling really sick. They're not really participating in social touching anymore. And so they can only really transmit by shedding the virus into the water column. So this disease trial setup, starting on this left-hand picture, basically we would infect a group of juvenile carp with the virus, and we would let them stay sick through 12 days of infection. And on each day, we would take one of those fish and move it into a larger tank with a group of uninfected carp. And then we would let them sit for 24 hours so that transmission could occur between those groups. And we would actually count the number of times that the infected carp would touch the other carp to measure the direct contact. And then we would move all those susceptible fish into their own tank and let them sit for a few days before we tested them to see whether transmission occurred. And we did the same thing with indirect transmission, except that we kept that those infected fish in a cage so that the only way they could actually interact with the other fish was through the water column, not actually direct contact. And what we found, starting on this left-hand picture, is that viral load increased kind of as we expected. It was a bit lower during the asymptomatic period, which was between day 3 and 6. And then the symptomatic period, when like those fish were actually looking very sick and acting differently, was between days 9 and 12, with sort of a peak of viral load at day 9 and day 10. Looking at the actual direct and indirect transmission, so we're looking at the number of contacts per hour. So this is how many times that vector fish was actually touching susceptibles with this black line. And you can see that during the asymptomatic period that contact is really high, and it drops off during the symptomatic period. The purple and green lines are showing the direct and indirect transmission groups again. And you can see that the percent transmission was actually really high during the asymptomatic period when there were a lot of contacts, and then it kind of drops off during the symptomatic period. There was very little indirect transmission, so indirect transmission is probably not very important even in wild settings when it comes to KHV infection. Another interesting thing that we found was at day 9, there was very little contact happening, but there was still a lot of transmission, which means that the viral load was also comes into play there. So maybe you don't have as many contacts, but you still have a really high viral load. So any contact you make is really effective in transmitting the virus. So again, this is really useful information for modeling this disease. And it's something that we just previously didn't know. We didn't have this information on how KHV was transmitted. So we also wanted to move our work out into the field to understand the ecology of KHV in wild populations. We do expect that demographics like the sex of fish, being male or female, or the age of fish could influence the impact of KHV on these populations. So we conducted a pretty intensive field study using boat electrofishing to catch carp. And then we would use a combination of different screening methods to identify different disease states, since it's not just as simple as infected and uninfected with KHV. There's different disease states that are present. So we could use PCR for detecting the virus itself. We could use the RT-PCR method to look for whether those infections are replicating or not, like we did in the disease trial. And we could also use ELISA. It's another type of test that looks for whether carp have actually been exposed to the virus in the past, even if we can't detect the virus present at the moment. So starting with this top left chart here, what we're actually looking at is the way that we interpreted those tests. So if you have a positive ELISA and positive PCR, that means that they were exposed a while ago, but they still have the virus, which means it's a persistent infection. If we can't detect the virus anymore, but we still detect antibodies to it, it means that they were exposed in the past, and now the virus is either suppressed to the point where we can't detect it, or it's gone, which is a latent infection. If they were didn't have any antibodies to the virus, but they do have the virus, that means it's a brand new infection that just started, so that's an acute infection. And then negative is just negative for both tests. So moving over to this right-hand panel up at the top, we're looking at the relationship between CARP age, which is proxied by CARP length, and different disease states. And what we found was that acute and mortality disease states were associated with younger fish. So basically, if you're a younger fish, you haven't participated in social sort of contact, you haven't participated in spawning. And so as you join the adult population, you're very likely to get sick and you're likely to die. But if you're an older fish, you're not likely to get sick and die from KHV anymore. You're much more resistant. We also found that the population was skewed towards females. So if you look at this bottom left panel, you would normally expect the male to female ratio to be about 50-50. But we found that there were significantly less males than expected. And also that these males were more likely to be associated with acute or mortality state. And this makes sense since male carp actually mature sooner than females, and they participate in spawning and social contact more at a younger age than females do. And then finally, looking at the ELISA values, you can kind of think of this as how resistant the carp are to KHV versus the age. So again, proxied by standard length. You can see that older carp have more resistance to KHV. So they have more of those serum antibodies, which explains why they're not as likely to have acute infections or to die from infection anymore. Also, we found some interesting things about seasonal prevalence and viral load of KHV in wild carp. So looking at this left-hand chart, what we're looking at here is the number of, or the proportion of fish that were positive for KHV in different tissues. And the different tissues are brain. We also looked at whether the brain infections were actually replicating or not. And then also gill and pink and kidney and gray. And what's interesting is that you can see that KHV seems to just kind of hang out in the brain. It's even there when there's no replication occurring. And it's also there when there's no other tissues that are positive for KHV. But that those other tissues seem to become positive for KHV later in the summer, so May, June, and July, with gills spiking in June, which means that's probably when most of the shedding is occurring, which makes sense because that's during the spawn. And then looking at this chart on the right is kind of looking at the same sort of relationship, but in a different way. So this is the viral load, so the actual number of viruses in the tissue versus the water temperature. And you can see that, the viral load in brain kind of stays pretty consistent throughout the summer, but these other tissues seem to sort of spike as water temperature increases. And this is sort of what we would expect, but it's really helpful to understand how the virus is actually working in this population. So the next steps for this project, we need to continue to investigate new and local outbreaks of KHV and CEV to keep putting dots on the map. If we're actually going to think about using this as an augmentative approach, we need to know where the virus already is and isn't, so we can kind of plan out how you would, potentially use it. We also need to refine our sampling and mortality estimation to get a better handle on how natural outbreaks actually impact these populations. And we want to isolate more strains of KHV so that we can do more detailed population genetics analysis, which can tell us more about how the KHV has been spread throughout the state. And also, if tools become available for CEV for isolation, we could maybe bring that virus through the same sort of research strategy. We also need to complete ongoing laboratory and field projects. So everything you're looking at here is really a sneak peek of what we've been working on, but there's a lot more samples that I need to analyze, a lot more testing I need to do, and a lot of analysis. With that increased information, we're hoping that will help us plan additional surveys and disease trials to gather whatever information is missing. And the idea is to incorporate this all together, this laboratory and population analysis of KHV disease dynamics into a disease model that includes management strategies. That way we can actually plan out like, hey, what are the different sort of handholds that we can have on this virus and on these populations that could feasibly work as a way to manage this virus for control of carp populations. And that includes, and then I guess the next step is really communication with all of you. So thank you for your time. I appreciate it. I want to thank everyone who's been involved with this project as well, and also our funding. And with that, I'm ready for questions.
All right. Thank you very much, Isaiah. Excellent presentation. A lot of information to get through there. So if you missed any of that, don't hesitate to drop your question in the chat box or the Q&A at the bottom of your screen, and Isaiah can answer them. All right, don't be shy. You can put some questions in the chat box there. Maybe Isaiah, I'll give you a question that we get a lot when presenting this project. What about lakes that are already infected? So a lesion already has KHV. Could this still be a strategy for biocontrol, or how would that work?
Yeah, so thinking about this from more of an augmentative biocontrol approach, I think that it's some of the lakes that interest me the most are the ones that already have the virus. We don't need to actually move it into those populations. And we do know that it tends to have an impact on younger fish. One of the major problems with controlling carp is controlling recruitment. So when you remove a bunch of adults from a population, you maybe don't get them all, and they can just spawn, and then the recruitment will bring a whole bunch of new carp into the system. But it looks like KHV really impacts those younger carp. And so it could actually be a really good strategy if you're able to manipulate the population, bring younger fish in contact with older fish before they can fully recruit into the population and replicate themselves. Maybe by a baiting strategy, which is already used in a lot of lakes right now. So corn baiting tends to really aggregate carp. And lakes that have sort of feeding stations, like in Blue Springs, Missouri, have had really significant die-offs because the population is so aggregated. So this is something that really makes me excited about potentially combining what we know about carp aggregation with what we know about KHV and CEV mortality.
Great. So we have a couple of questions coming in here. Here's one from Heidi. She's curious about the potential for KHV to mutate and potentially impact native fish, even though it doesn't seem to do that right now.
Yeah, so there's definitely some evidence of herpes viruses in fish potentially like jumping hosts in the past, but this is really on the scale of like thousands or millions of years. KHV is a herpes virus, which means that it's a DNA virus with a huge genome, so it doesn't really like to change. It really is well adapted to carp. So what we find more and more of the more that we look at this virus is that it is really focused on carp. It doesn't seem to infect any other fish. And definitely doesn't cause any disease in any other fish. And its evolutionary rate is extremely low. So that's something that makes it really exciting to find KHV in particular, since it seems to be a much more slowly evolving pathogen. Of course, it's always possible that a virus can mutate, but this isn't like the flu where it's like a segmented RNA virus that is capable of infecting a whole bunch of different hosts and recombining in them. That's very, very different. It's sort of similar to some of the comparisons between even coronavirus and the flu. People are worried about coronavirus mutating, but it's not. It just doesn't have the same evolutionary rate as the flu does, and so we don't worry about that as much. And KHV is even much more stable on a whole different scale than those viruses are. Those are RNA viruses.
That's a good response. And maybe I would just add, Isaiah, that if that concern is still too high for some people, the fact that KHV is already here and this augmentative strategy is a fairly low risk activity, since it's already presented with the opportunity to expose native species.
Yeah, another way to look at that too is that these populations are extremely dense of carp. So, if we're worried about viruses mutating in carp and causing disease in other species, it's even more of a reason to think about trying to remove carp sooner rather than later, because the virus is there and it's here to stay. Probably CEV as well. And so we really don't want to, if we're worried about viruses moving around, these are just ones we know about. There's a whole slew of other pathogens that are out there that these dense carp populations could be acting as a sort of vector for.
All right, we have a question here from Jeff. He's wondering if KHV only infects common carp, or could it also affect silver and bighead?
It only infects common carp, so it doesn't even affect other cyprinid species like the ones you mentioned. Common carp is Cyprinus carpio, and those other species that you mentioned are totally different, and KHV doesn't impact either of them.
Okay, we have a question here from Eric. I think you touched on this, Isaiah, but just to reiterate, he's been hearing a lot about herd immunity these days. Will carp eventually build resistance to the virus, and what might that look like over time in a population?
Yeah, so even though I didn't actually use the words herd immunity, what I was showing you with that ELISA data and the, let's see if we can get back to, I skipped way past it. So what we're actually looking at here is not like necessarily a measure of herd immunity, but it is similar. So all of these fish that have detectable levels of anti-KHV antibodies are actually sort of that herd that is immune to KHV. But what's interesting about KHV is that just because, and with fish diseases, is that just because you have antibodies to it doesn't mean you can't spread it anymore. So not only are these fish immune to the virus, which means that we can't kill them with KHV, they have to be taken out with a different method. They're a sitting reservoir of KHV that can infect the younger population that doesn't have immunity yet. So it's kind of like a double-edged sword here. Like, yeah, you can't get rid of the really immune adults with this removal method, but adults are really susceptible to some of those other methods like the seining, electrofishing, methods, while the juveniles are the ones that we really have trouble controlling. So this might be something that if we kind of balance those two things, keeping the right number of adults in the population, maybe we can actually reduce recruitment using those adults.
Great. Just a reminder, you can put your questions in the chat box or Q&A. We have a question here from Kristen. She's wondering what she should do if she sees a fish kill, a carp mortality event.
Yeah, so if you see a fish kill, which is like, even if you just see like 5 dead carp kind of floating in the water or on the edge of a lake, you should report that to the DNR duty officer. And they've been really great about communicating with us and letting us know when those fish kills happen. And also you can check out our website. We have a link to a fish kill reporting app. And you can actually just drop a pin right on your lake and it'll notify Nick and myself right away, and I can actually go out and check out those fish. I've still been doing that really actively and have diagnosed a number of carpedema virus fish kills this last year. And so keep reporting those. It's a really great way to keep learning about where the virus already is.
Yeah, it's a good point. I just put a link to the fish kill app there in the chat box if you're curious to check it out. All right, so we've cleared through the questions that we have right now. All right, another question, Isaiah. Do you have any ideas of how much it would cost to implement this in a lake, including perhaps the cleanup of the dead fish?
So usually during these mortality events, there's no cleanup of the fish. They just tend to rot over the course of a week, and then they're gone. So they don't stick around too long, which is a good thing for that prospect of having to clean them up. But in terms of cost, I'm not really sure how much it would cost to do this. It's just not something that we really know how we would target it. How we would introduce the virus yet. It would probably have to be with infected fish, or we're just looking at a lake that was already infected. I think that the cost could be really, really low. When you think about a lake that's already infected, then you could be looking at just doing sort of like normal removal methods like aggregation. So it would just be whatever CARP Solutions or WSP charges for their removal methods.
All right, well, just.
I will say one more thing about that though. In general, one of the things that people really like about biocontrol methods is they tend to be a lot cheaper than other control methods. I was just talking with someone yesterday, Justin Townsend, who has had some success controlling Eurasian water milfoil with a weevil, and really they get a permit, they go collect those weevils, they bring them to the lake, they drop them in there, and then they kind of work to keep that population going after they've introduced it, and they've had a lot of success with that, and it's been a lot cheaper than other methods that they've had. A lot of people are turning to biocontrol methods because they're using their control dollars in other ways, and they're looking for cheaper methods. So biocontrol tends to be a lot cheaper, and I don't think that it would be any different for KHV and CEV.
Great. Well, we don't have any more questions right now. We can wait another 30 seconds and see what folks come up with, if they do. We had a question here from Joe. Who was doing the weevil work?
That was Justin Townsend from Ramsey County AIS. Sorry, I forget his job title. But do you want to jump in and tell everyone his job title?
Yeah, I believe he's the AIS coordinator.
AIS coordinator, yeah.
Yeah, there's a few groups around the state right now doing experimenting with weevil control. All right, well, we will go ahead and end there. I encourage everybody to check out the next round of presentations starting at 1 o'clock. And virtual round of applause for you as an excellent presentation. Thank you very much.
All right, thanks everybody.